In chapter 41, we are going to discuss about the biotechnology and the industrial microbiology. Now, one of the important part is that many of these microorganisms do produce a lot of different things, and exploiting these microorganisms is absolutely necessary for producing many of these pharmaceutical products, many of these antibiotics, a lot of different things. And we are going to look at all these different industrial material which are industrially important. And we are going to look at that why these industrial microbiology is such an important field, because it touches our life on everyday basis. So let's take a look at these some of these major products of the industrial microbiology. So, of course, we know that the ethanol is produced for many different purposes, and one of the yeast is, although there are different ones, but one of these is a Saccharomyces cerevisiae, which is the baker's yeast. That we have discussed in the last chapter. Then acetone butanol, so many applications, is produced by this Clostridium acetobutylicum. Butanediol, this is also butanediol, is also produced by Enterobacter serratia. Then amino acid, say for an example, lysine is a bread additive. We use the glutamate, which is a part of the monosodium glutamate. Now, some people use it, some people don't, but that's also one of the product. There are many different amino acids which are produced by this Corynebacterium glutamicum. Many different types of organic acid, like, for example, citric acid, acetic acid, lactic acid. All of these are produced by different microorganisms. Here, the citric acid is produced by a fungi, Aspergillus niger. Many of the vitamins are produced. As you can see, polysaccharides are also produced. And say, for an example, these bioproduced dextrans are used. And these polysaccharides are the part of many food additives. And you will see that many of the times there are lots of these different polysaccharides for the consistency of the jam, jellies, and all those kind of things. They are added. Xanthomonas produce those. And antibiotics, of course, penicillium, most of the antibiotic are produced by the Streptomyces, also Bacillus produce some. And interestingly, this alkaloid, which is produced by the Claviceps purpurea, and we already know that they actually produced some of these mycotoxins, which are actually lethal. So what are the applications of these alkaloids? Now, these alkaloids are produced by this Claviceps purpurea, and then they are processed, and then they are tagged with some drug delivery system, and these are used to target the cancerous cells, where particularly these alkaloids or the toxins will go to the cancerous cells in the tissues, and basically they will have these detrimental effect where they produce these planar compounds, and these planar compounds intercalate with the DNA and basically hypermutate the cells, and essentially they will die. Then steroid transformation, Rhizopus arthrobacter. Then there are many of these hormones like insulin, human growth hormone, somatostatin. These are the different hormones that are produced by recombinant organisms. Now here, one of the most important part is that in industrial microbiology, there are many products which are the natural products, and sometimes they are produced by a wild type, or they are mutants or a selected strain variety. On the other hand, where we are looking at particularly this genetic engineering, this part, these products are not natural product of the bacteria. These are the natural product of the bacteria. We just control the environment to produce more of it for our benefit. But on the other hand, these interferon, somatostatin, insulin, human growth hormone, these are not produced by the bacteria, but these are the recombinant DNA products. So basically, the gene is cloned into either E. coli, very good choice, or in an yeast, which is also can be grown in large bioreactors, which are where these recombinant proteins can be produced and isolated. Even there are some of the vaccines which are actually produced by this way. So the other one is biofuel, hydrogen. We all know that there is a problem with the fossil fuel. At one point of time, it will run out. So these are some of the alternative fuel, like hydrogen can be produced by some of the photosynthetic microorganisms. Good choice. We're going to talk about that. Methane can be used as a alternative fuel as well. And ethanol can be produced by some of these bacteria like Xybomonas or the Thermoanaerobacter. So you can see that these are just a handful of example of the products that we use in industrial microbiology. There are many, many more, and this list is by no means even close to complete. So let's take a look at that, how we developed or produced some of these products. So the first one is the antibiotic. And interestingly, that about 65% of all the antibiotics are produced by the streptomyces. And the antibiotics are secreted by the microbes and in the native states, and sometimes they are chemically modified. So most of these antibiotics, as you can see, that they are produced by the streptomyces, but also the penicillium, we are looking at this example, is produced by Penicillium chrysogenum. And what it requires, that it requires a careful, precise control of the media to yield the maximum product. So let's take a look at that how we basically maximize in a bioreactor the production of penicillin by Penicillium chrysogenum. So here at the initial period, you can see that the main substrate for that is lactose. So the fungi, Penicillium, uses lactose, and over the period of time, the amount of lactose actually goes down. So you can see that the amount of lactose goes down. And at that time, like you can see that this phase is the biomass growing in a logarithmic phase. This is the logarithmic phase. On the other hand, you can see that right here is the nitrogen, and over the period of time, you can see that the nitrogen, ammonia, that level is also going down over the period of time. So what happens that when the lactose is going down, as well as the nitrogen content of the media is going down, at that point, the bacteria actually think that it is under a stressed condition. And the stress condition actually trigger the highest amount of penicillin production, because remember that what is the purpose of an antibiotic? It is basically to kill other organisms and having advantage of the environment. So this is basically a stress related product where there is a competition. It will have a highest amount of production of the product, which is penicillin. So at this time, what happens that the penicillin starts, the amount of penicillin actually grows up. But at that time, as the lactose is depleting over the period of time, and as well as the ammonia, we need to feed them with the glucose and the nitrogen so that they survive. They go into a kind of a stationary phase, but they do not die. But see right here, you can just initially add 1.45 gram per liter glucose. Then you just reduce that to 1.31, 1.15. So you're reducing that, and you just in this phase pretty much you use about 18 milligram per liter of nitrogen feeding. And you can see by these controlling these over the time temporarily, when you control these amounts, you are getting the highest amount of penicillin production. And you can just appreciate that penicillin is not produced in the highest amount in the logarithmic phase of growth, but it is actually produced in kind of like almost where you have close to the stationary phase. And this is basically a stressed condition. And at that time, penicillin is produced at the most. Sometimes so this is the production of penicillin in a bioreactor by Penicillium. Now, there are many modifications of the penicillin. Say, for an example, penicillin G is produced when their phenyl acetic acid is added to the media, which has a benzyl side chain, and this benzyl side chain is added. And usually the fermentation takes about a week, and the broth is separated from the fungal hyphae. And then the the antibiotic is basically purified, crystallized, and then these antibiotics are modified by the chemist. Say, for example, amoxicillin, ampicillin, then methicillin, piperacillin. There are so many derivatives of the penicillin. These are all essentially modification of the chemist. So essentially what happens, the basic beta-lactam ring, the squarish ring with a 5 carbon associated with that, this beta-lactam ring, that structure, original structure of the penicillin remain the same, but addition of many of these side chain actually do have many applications, shelf life increase, as well as a broad spectrum activity that we have talked about in the other chapter. So let's take a look at some other materials. So the amino acids are used in the food industry as nutritional supplements in bread products, flavor enhancing compound. So lysine is actually used as the in the bread as an addition, and we already talked about the glutamate or the monosodium glutamate, although the usage of that is to some extent controversial, but not going into that. But these are some of the product that is used. So let's take a look at that, how we basically produce these. So the glutamate is produced by a bacteria, Corynebacterium glutamicum, and a carefully chosen strain actually is used where this bacteria does have a mutation where when it is actually working with the Krebs cycle or the TCA cycle, they cannot convert the alpha ketoglutarate to the succinyl-CoA. So as this basically this pathway is blocked, there is a separate pathway that it bypasses the TCA pathway to produce glyoxylate, to produce malate, and malate goes back to the acetic acid, and that's how it actually survived. But the interesting part is that when the alpha ketoglutarate is accumulated with the help of the glutamate dehydrogenase, this is essentially converted to the glutamate, and this is what is basically secreted into media. But what happens that the glutamate actually is produced in the stationary phase. So initially what happens when at the highest amount of the growth of the Corynebacterium is achieved in the log exponential phase, where the isocitrate, then glyoxylate to malate, oxaloacetate, this cycle actually is allowed to grow, and this will basically grow, make the ATPs for the bacteria, and you have enough amount of bacteria in the bioreactor. And then what happens that at the stationary phase, when the nutrients are depleted, at that time, almost a molar conversion of isocitrate to glutamate actually essentially occurs. And there are some of the specific things that are controlled. So the biotin levels are kept low. And then also the addition of fatty acid derivatives increase the membrane permeability of the bacteria, and glutamate is basically released into the broth or in the media. So this is how glutamate is manufactured, and which is also one of the amino acids. And there are similar pathway for many of these amino acids. Just varies in the specific steps, but essentially they are all the same. On the other hand, lots of organic acids are also used, commonly used as preservatives and also flavoring agent. Citric acid does have this tart flavor, acetic acid, so many types of vinegar, so many different types of vinegars are produced or for different application, flavor enhancing agent. Lactic acid is also for preservation, as well as making, like say for example, making sauerkraut. These are all different flavoring agent and preservatives. So let's take a look at that, how commonly, say for example, citric acid is produced. So citric acid is produced by fungi, which is the Aspergillus niger. And initially what happens, it is grown in a high sugar concentration, 15 to 18%. And limiting the trace metal like manganese and iron to stop the fungal growth, essentially. So the regulation of the glycolytic pathway and the TCA cycle, and at that time, the citrate synthesis activity actually increased at that time. So what happens that the first step from oxaloacetate, the first citrate synthesis is in the Krebs cycle. The first step from oxaloacetate, citrate synthesis, produce the citric acid, and you want to stop the reaction at that point. And that is essentially controlled by the stressed microorganism with high sugar and manganese and iron to stop the fungal growth. And at that time, the next step in the say, for example, the next step in the TCA cycle, which is involved the enzyme aconitase and isocitrate dehydrogenase, their activity actually decrease. So basically what we are doing, that we are stopping just the same way that alpha ketoglutarate but we already saw that how we basically manipulate the production of the amino acid. The same way here, as the next step of the citrate synthetase or the citric acid formation is inhibited, the citric acid accumulation happens in the stressed microorganism. So this is basically the production of these one of the organic acid. And the same way, there are many different the lactic acid as well as the acetic acids are also produced just by different mechanisms. So after that, we are going to take a look at the industrial enzyme, and many of these industrial enzymes are absolutely useful in the pharmaceutical, agricultural industry, food industry, consumer products, and it's about $5 billion annual cost of these, that's basically a $5 billion industry essentially. And these enzymes are of a bacterial and fungal origin, and we call this as a biocatalyst. So the advantage of these biocatalysts are their reduced cost, and we do not have to use the chemicals as for the catalysis, and they are less toxic as well. So so these are essentially, so you can just see that they can break down natural polymers like starches and the proteins, and they essentially provides a consistent quality, and most processed foods have been treated by at least one enzyme. So let's take so one of these is, for an example, one of the industrial product of these enzymes is a pectinase. So the pectinase is usually the fruits or where the juices are extracted, like citrus fruit. They are say apple or oranges, these are treated with a pectinase, and that increases the juice recovery. So that is one of the application. Say, for example, that in the last chapter, in chapter 40, we talked about the rennin. So the rennin is essentially extracted from the animal stomach, and that is essential for the cheese industry to coagulate the milk. So this is, on the other hand, an animal stomach source is the rennin. And in the rennin, the chymosin is an active product. And so that is the enzyme specifically. Rennin is basically a mixture of different enzymes, and chymosin is one. And now the recombinant bacteria, by the genetic engineering, the chymosin gene is essentially put into the recombinant bacteria, and industrially these chymosins are produced from the genetically modified microorganisms. On the other hand, another example is a lactase. So many are allergic to the lactose or lactose intolerance. We know that, and that's the reason that there are milk that are sold which are lactose free, and they are treated with the lactase to remove the lactose from the dairy product. And all of these enzymes actually come from the microbial origin. On the other hand, the last one that what we are talking about is the laundry and the dishwasher detergent. And these rely on enzymes to remove the stains, blood, and grease. And to remove them, we usually use the different phosphates and their derivatives, but what happens that these are released into the environment and they can accumulate into the water bodies, and that cause eutrophication. Yeah, and this eutrophication is essentially that high amount of phosphates in the water bodies which actually enhance the algal bloom. And you can see this like all these green ponds and all those kind of things, and this is these are some of the problems in the environment. So if we can replace these phosphate-based detergents by proteases and the lipases, the proteases and the lipases can actually digest many of these proteinaceous and lipidic grease blood stains, and those are much more environmental friendly. So next we can talk about the vaccine, and again we are essentially not talking about the different types of vaccine that we actually talked about, but here we are talking about that what are the different approaches in the biotechnological standpoint where new types of vaccine can be developed by different mechanisms. Say, for an example, the most important is that the reverse vaccinology, where you mine the genome sequence of the pathogen, assemble a list of antigens, and a good vaccine target. So again, one of the important part is that when the antigen is essentially the antigen target must be expressed by a pathogen during the infection, and be secreted on the surface of the pathogen, found in all strains of the pathogen, and elicit a host immune response, and essential for the pathogen survival. So these are some of the target for these antigens. Say, for an example, on the other hand, say there is one of these reverse vaccinology process that was used particularly for the Neisseria meningitis. Neisseria meningitis actually cause meningitis, and there are 5 strains of that. It is A, C, B, it is A, B, C, and WY. So it is 5 strains are A, B, C, and WY. Now, very interestingly, there is a vaccine that was available for a long time for the strains A, C, and WY. But for the, and essentially the vaccine for the ACWY is made from the capsular polysaccharide of these 4 serovars. And unfortunately, what happened, the same polysaccharide of the serovar B, which is Neisseria meningitis type B serovar, that polysaccharide is found on a human cell. And that's the reason the antibodies essentially can be raised and they can attack the human cell itself. So that was not a good target. So by the process of reverse vaccinology that was used, the genomic sequence of the entire serovar B was deduced out. And after mining, the antigen was finally found. And in that process, essentially, initially about 600 surface proteins were considered for this serovar V. Then about 300 was tested on the mice, and 25 were worthy for further study. And out of those, there was some antigen that was basically identified, which does have all these different properties expressed on the pathogen surface of those, all strains of the pathogen, elicit a home immune response is essential. And then in 2014, this vaccine, it came into Europe a few years before, but in 2014, it was licensed in United States. Now we have the vaccine of the meningitis ACWY as well as meningitis B together. So it is now, you can be protected by that vaccine for all the 5 serovars of the Neisseria meningitis. Now, one of the drawback that earlier had, and that was not an issue, see right here, it needs to elicit a host immune response. Now, being a capsular polysaccharide, similarly, they do not actually produce a good amount of immune response. So all these polysaccharides are usually conjugated by a diphtheria toxin, which is a diphtheria toxoid conjugate. And when these polysaccharides are usually conjugated with the diphtheria toxin toxoid, that elicit a good immune response. And essentially, the vaccine is very effective against all 5 serovirus of the Neisseria meningitidis. So this is also another type of vaccine discovery. So essentially, the way we develop that we basically see some of the recovered patients from a disease, and we can isolate and ask them to donate their blood, and we can isolate the B cells which are generating antibodies specific to these pathogens. And then what happens that after that we can create the cell lines which can produce these monoclonal antibodies, and by the hybridoma technology. So from the B cells are fused with a myeloma cell, and the technology we are going to learn in the cell biology. It's beyond the scope of this, but these hybridomas actually produce these monoclonal antibodies, and we use computer modeling to identify the target of each monoclonal antibody on the pathogen. So now we know that where is the epitope of the pathogen, and this process is known as an epitope mapping. By epitope mapping, we know each monoclonal antibody is binding to a target, which is this antigen. And then we design a peptide that mimics the immunogenic portion of the pathogen, and we can artificially produce that peptide, and then that peptide can be injected to the human being just as a protein. It's a piece of peptide and protein, and that is essentially the antigen. And that's how we can formulate a vaccine to immunize the susceptible individual. Now, it is not that easy, because remember that a protein is a larger structure, and it three-dimensionally folds in a certain way. When you produce a smaller peptide, sometimes it will not fold the same way, and that may not be recognized by the monoclonal antibody. So there are some modification of these steps. But essentially, this is what is called the peptidomimetic antigen. So it will just basically mimic that. It needs a lot of permutation and combinations, but this is also the way that vaccines can be discovered. In the next section, we are going to compare the biofuel and discuss the typical type of biofuel. Now, why this is important? And as we can see, the population and economic growth increases the energy demand, and the populations are growing at an alarming level, and the fossil fuel that we have, that is depleting fast. And at one point of time, may not be in our lifetime, but at one point of time, that the fossil fuel energy will deplete. So we have to have some kind of alternative energy. And these are the reason that many of these biotechnology companies and the policies and processes are there to have alternative energy. And one of these is the microbial energy conversion is used in this process, where the microbial transformation of organic materials into biofuel, which is essentially one of these two of these most attractive biofuels are the ethanol and the hydrogen. So let's take a look at that. What are the pros and cons of production of ethanol and the usage of ethanol? So ethanol is currently used as an additive to the gasoline because it's an easy to store, can be burned in cars designated to use pure gasoline. So most of these pump gases are usually used about 10% of the ethanol. We call this E10. There are some cars which can use E85, 85% ethanol. So those are some of the modified engines that are also there, but that's not a mainstream. But let's first talk about that, how they are produced. So ethanol is produced from the degradation of plant starches and by the amylases and amyloglucosidases. In US, the main source for these starch is corn, whereas the sugarcane and palm is used for this source in other countries. So when the amylases and amyloglucosidase essentially break down the plant starch, result in the sugar, in the glucose, and this is essentially fermented to ethanol. But what happens that when you used, say for example, corn, that also elevate the cost for pesticide and usage of fertilizer, and that elevates the cost of food as well. On the other hand, the main thing that are now being essentially developed are the crop residues, and these are left in the field after a harvest, and there are some of the grasses called, like say for example switch grasses, which which grow essentially in the lands which are not fertile for agricultural practice. So crops will not grow, but the switchgrass can grow, and these could be the sources for the cellulose or the hemicellulose. So essentially what happens, the cellulose and the hemicellulose are polymers of different hexoses and pentoses. And what are those? These are 5 different types of hexoses and pentoses. These are the glucose, xylose, mannose, arabinose, and galactose. So these again, glucose, xylose, mannose, arabinose, and galactose. And these are the sources of the sugar. Now, the cellulose is degraded usually in the industry by heating with acid about 100 degrees Celsius, which is an extremely harsh and a corrosive condition. And of course, energy intensive for heating up that as well. So now the microorganisms are being engineered that can degrade the cellulose as well. So you do not have to use the acid, but you still have to use that temperature 100 degrees Celsius plus. And these microorganisms are thermophilic or hyperthermophilic, which can grow at 100 degrees Celsius as well. So the interesting part is that although the cellulose, hemicellulose basically is broken down to glucose, xylose, mannose, arabinose, and galactose, the only glucose can be converted to the ethanol. Still the xylose, mannose, arabinose, and galactose, this cannot be used to produce. So the scientists are basically in lookout for microorganisms which may use the xylose, mannose, arabinose, and the galactose, which are the products of the cellulose and the hemicellulose, if they can be converted into some pathway to produce glucose, and that through that we can produce ethanol from those sources as well. So that will increase the cost effectiveness. So, but what happens that the ethanol does have some disadvantages. The ethanol actually absorbs water, and this cannot be shipped through the existing pipeline that's the reason that because the condensation will happen inside the pipe for that. And that's one of the reasons that it must be distilled twice to remove the water, one at the source and one at the delivery. And that is a huge problem. And also another important thing is that it contains less energy than other fuels. And that's one of the basic research that what is going on, that the researchers are trying to develop microbes that produce different types of alcohols, like in isoprenoids and other alcohols and other hydrocarbons, which can closely resemble the gasoline, which does have low hygroscopy and high energy density, and also maintain the fluidity in the cold. So these processes for artificial gasoline is in the process, but yet that is not a mainstream. But at one point of time, this technology probably will be coming to exist. So now the next biofuel is essentially the hydrogen gas production. And the hydrogen can contain 3 times more potential energy per unit than the gasoline. And this is the highest energy content fuel available. And you can just easily understand that the rocket fuel is basically hydrogen and the oxidizer as an oxygen. So hydrogen and oxygen is so hydrogen is essentially the fuel for a rocket. And there is a reason for that. It's the highest energy content fuel available. And say, for example, NASA actually uses hydrogen fuel cells, very common as well. And the Problem is that hydrogen can be mixed with gasoline, unlike ethanol, to reduce the petroleum use. It has to be used as a gas, which could be pressurized, which could be liquidated, but it cannot be used as mixed with other fuels as well. So hydrogen production is essentially a product of fermentation. And there could be two different types. It is not only an anaerobic process, but could be an oxygenic process as well. But there is a catch in there. The photosynthetic oxygenic microbes like the cyanobacteria and algae, they can produce these and two of these most important enzymes that is related to that is the hydrogenases and the nitrogenases, and both of these are actually required in this process. Now, the problem is that these are sensitive to oxygen, and that's the reason that we talked about this, that part of the process is an anaerobic process. So just remember that although the oxygenic photosynthesis, that cyanobacteria algae actually happens, the part of the cell where the nitrogenases are at that time, they are not exposed to the oxygen. And usually the oxygenic photosynthetic microbe produce this hydrogen during the nighttime. But there are some problems with this. So oxygenic photosynthetic microbes essentially will oxidize this polybeta hydroxybutyrate, and they produce a lot of hydrogen. But the problem is that this hydrogen from these oxygenic photosynthetic microbes must be removed fast, because production of the hydrogen, that is inhibitory for the hydrogenase is inhibited by the hydrogen gas. And that is one of the problem, that immediately it has to be removed, and there are some of the process to make the process cleaner and more efficient overall. So that is one of the problem. In contrast, on the other hand, there is also another type of hydrogen production, which is the anoxygenic heterotrophic bacteria that use the cellular hydrogenases and the nitrogenases. And this is of interest because what you can do, that the atmosphere where this bacteria is growing, that can be purged out all nitrogen. And at that time, the H2 will form from the nitrogenases. And the reason for this process is that hydrogenase is basically inhibited by the hydrogen gas, but the nitrogenase is not inhibited by the hydrogen. So in this process, as the nitrogenase is producing the hydrogen, the problem with the oxygenic photosynthetic microbe usage is eliminated. But still, this process needs a lot of ATP. But the electron conversion almost can reach to 100%. And essentially, what we essentially think that at one point of time, it will be some time, but eventually the fossil fuel will deplete, and we may at one point of time see that the gasoline could be produced in bioreactors or alternative fuel for in some form at one point of time. That will be probably the future. Now we are looking at some of these microbes that can be grown in the industrial setting, and they can essentially pose some challenges. And we are going to talk about the fermenter and where it is used, and the two types of fermenters and what they are. Now, when we talk about the word fermentation, that is interesting, that this is a part of the cellular energy conversion process where the pyruvate that can be fermented into ethanol, lactic acid, or acetic acid. These pathways are known as the fermentation, essentially. But when we use the word fermentation in an industry, that is essentially defined in a different way. So in the industry, the fermentation means the mass culture of the microorganism, not necessarily that it's only the production of the ethanol, lactic acid, or acetic acid. And we basically developed appropriate media and conditions that is critical for the producing the microbial products. Now, when a particular product that need to be produced is identified, that is basically studied in the laboratory, in the microenvironment, in small culture, and you just develop that over the period of time. And once that is done in a very minuscule scale in the laboratory, you gradually scale up that fermentation to the large fermentation. Say, for an example, a 500 ml bioreactor is probably starting a culture in the laboratory, from where it can go to a 3 liter, 6 to 10 liter, 2,500 and 1,000 and 100,000 liter scale up large fermenters. But still, in the laboratory of these industries, a microenvironment is used for a small culture to keep a pure culture as a backup. By chance, if there is a failure of the large culture in the fermenter, something happens, so that they always have the backup pure culture as well. So let's take a look at these fermenters. So this one is basically a picture of a stirred fermenter, where you have these impellers. So let's take a look at the structure of this, and then we will talk about the different other things about. And this fermenter can be run under an oxic or anoxic condition. So here you can see that their air is in, and if it is an oxic condition, the air will be purged, but in an anoxic condition, you will not have that. So here the culture nutrient are added. These are the motors that will run the impeller to mix it up. And essentially, this is the sample line that can be that is added, and you have a cooling jacket around the fermenter, and there are several different sensors. You can see that you have temperature sensor control unit, you have a pH probe, dissolved oxygen probe, biosensor unit, which can measure essentially the product. So all these things are essentially in there. This is the harvest line where the product can be removed from the bioreactor. So these nutrient addition, sampling, fermentation monitoring, these can all carried out under the aseptic conditions. And essentially the biosensors can give you the real-time information about the course of fermentation and specific substrate, metabolic intermediate, and the final products can be detected by these biosensor unit as well. Now, this is called a stirred fermenter. And here, continuous addition of the critical nutrient is so that the microbes will have an excess substrate at any given time. And this prevents the production and accumulation of undesirable metabolic waste products. And in these type of fermentations, which are not batch fermentation, these are continuous fermentations, and this is essentially this is where the bacteria is maintained essentially, or the fungus is essentially made in their exponential or log phase. Remember that at the very first time we talked about the penicillium production by and we talked about that, but that type of fermentation is a little bit different. That is essentially a batch fermentation, so everything is put together. There are a couple of things that are monitored, but that fermentation actually goes on for 7 days, and after that 7 days, the entire batch is harvested, cleared. The entire fermenter is cleaned up, and a new batch is put for another 7 days of culture. So those are batch fermentation. Whereas here we are looking at a continuous feed fermentation, where you do not have to stop the fermentation, where you continuously feed the line, and you continuously harvest from this line as well. And you maintain the bacteria in the exponential phase of growth by adding continuous nutrient and excess substrate available, and you do not have accumulation of metabolic waste products. The other type of fermentation, which is not shown in here, is known as an airlift fermenter. So what is an airlift fermenter? That also purges air from the bottom like this, but the only thing is that it doesn't have an impeller. Now, there are some of the large, very large fermenters, 100,000 liter, million liter fermenters, where if you stir them with usually these impellers, it builds up too much heat essentially, and that can cause problem to cool the material down. So very large fermenters are usually airlift fermenters, and just the bubbling of the purged air can mix these the entire content of the media in a few seconds. So for example, just interestingly, that the largest fermenter, single vessel fermenter that is present in the world, the largest one, it is in UK, and it's the place called Billingsby, and this fermenter is 200 feet tall and 25 feet wide, and it's an airlift fermenter. That's the largest fermenter, single vessel fermenter that is present in there. Very interestingly, I was just looking in the news lately, and in 2019 the DuPont actually set up a probiotic fermentation facility in Rochester, New York. And at this time, that facility, although the industry does not say that exactly the volume of the fermenter, but essentially what is considered that DuPont facility probiotic fermenter is the largest probiotic fermenter in the world as well, which is based in the DuPont plant in Rochester, New York. So these are some of the interesting facts about the fermenters. So these fermenters actually produce these primary metabolites and the secondary metabolites. And what are these? The primary metabolites are related to the synthesis of microbial cells during the exponential growth. This will include the amino acid, nucleotides, end products, and the enzymes, essentially. On the other hand, the secondary metabolites are synthesized when the nutrients become limited or waste products accumulate following the active growth phase. So when we talked about these continuous fermenters in the last part, those are used to produce the primary. If the primary metabolites are the things that are being produced, you use a continuous feed fermenter to maintain the culture in the logarithmic phase or the exponential phase. On the other hand, if you just use the secondary metabolites, like synthesized when the nutrients become depleted, sometimes considered as a part of stress response, like the antibiotic production, you usually, what you do is basically a batch fermentation. It takes much longer time, but essentially you do not maintain the culture continuously in the exponential phase. You let it go into the stationary phase, which is essentially the stress response. You feed them with the nitrogen or the glucose just enough to keep them alive, but under seriously stressed condition. And at that time, the product is produced in highest amount, essentially. Next, what we are going to talk about is agricultural biotechnology. And we are going to talk about a plant pathogen, which is known as Agrobacterium tumefaciens. And this is a plant pathogen which causes a tumor in the plant, which is known as the crown gall disease. And we are going to talk about that, how this is used for genetic modification of the plants. And then we're going to talk about a pesticide, which is the Bt toxin. This is also derived from a bacteria, Bacillus thuringiensis, and we are going to talk about its characters and others. So the Agrobacterium tumefaciens is basically we use a plasmid that is present in this bacteria for genetic engineering in plants. So what is the process? So the process is that this bacteria do have a plasmid which is known as a TI plasmid, which is a tumor indensing plasmid. And next to that, there is a sequence of DNA, which is a tDNA. And this tDNA is a tumor-causing DNA. And there are some direct repeats on each side of this tDNA. And this allows the tDNA or the tumor DNA from the plasmid of the bacteria to be integrated into the plant's chromosome. And it is stably maintained. So the scientist actually took this TI plasmid and removed the tDNA and then transferred some of the typical characters of a recombinant vector or a plasmid vector, like multiple cloning site, a selectable marker with an antibiotic, and those things. These things are transferred into a recombinant plasmid. Now, in place of the tDNA, you can insert or clone a gene of your interest that you want to be stably expressed, or expressed means like it is integrated into the genome of the plant and it is stably expressed. And this will be a genetically modified plant, or basically we call this as GMO, genetically modified organism. And there are a wide variety of things that can be done in this process. So on the other hand, we are also going to talk about these bioinsecticides and the biopesticides, and these are important for the killing of many of these insects, which actually are responsible for huge crop losses as well. And 75% of derived from a bacteria known as the Bacillus thuringiensis, and it's known as the Bt toxin. We're going to talk about that. So first, let's take a look at that, how this bacterial Agrobacterium tumefaciens and the TI plasmid actually work. So what we do is so right here we have the Agrobacterium, and it contains the TI plasmid. Now we have another bacteria where we have our clone gene, and this is the gene of interest that we plan to put into the plant. And first it is cloned into another vector, a plasmid vector, from where it is actually cut out. And then the isolated TI plasmid this gene is inserted into its cloning site. And now this is a recombinant TI plasmid. So now this TI plasmid is returned back by transformation of the Agrobacterium. So now the Agrobacterium tumefaciens, which does have the ability to infect the plants, does have the recombinant plasmid. Now this is infected to the plants, and the plant cells will be infected by the Agrobacterium and by the homologous recombinations, because it contains these repeats, direct repeats on each side of the gene. There will be the integration of the gene into the plant chromosomes. And then the plant cells are actually checked for whether they have received this modification or not. So all the recombinant plant cells are actually isolated, and the entire plant is grown from the single cell. And how this is done, usually this is the tissue culture technique, from where you can just grow the plant cell just like the animal cell. From animal cell, you cannot grow an entire animal. It's not possible anymore. But from a plant, you actually do the tissue culture, which is basically they form into a callus. And then by the induction of many hormones and other things, they can be grown into the plants. And we call this as a plant tissue culture, very commonly done in all the agricultural biotechnology processes. And now the recombinant plant, when it will be formed, all the offsprings from there, you do not have to do the tissue culture all the time. All the recombinants that can be formed from the seeds of that plant will be the recombinant. And this is how you generate a genetically modified transgenic plant of your interest. And these are a variety of things that increase the shelf life of the fruits and all those kind of things that is also possible. Interestingly, what we are going to see that in the next part of our talk, we are going to talk about a toxin that is produced as a pesticide, where a bacteria produce this pesticide, which is the Bt toxin, and this is produced in industrial scale from the bacteria and isolated, purified, and used. Interestingly, there are transgenic crops that are developed by this Agrobacterium, where the Bt toxin is the gene, the toxin gene is inserted into the TI plasmid, and the plant does have now expressing the Bt toxin genes. And the classic example for these will be an example of a particular insect resistant plants. And there are several crops that are out in the market, which are the transgenic crops with insect resistance. What are the other applications? Very interestingly, what we are going to see that, say for an example, the influenza vaccine that used to be classically made into eggs. Like you just take these eggs, and there is a robotized process where you just make a tiny hole on the top of the eggshell, and you just put these viruses to produce to infect those, and essentially these antigens are essentially grown on these viruses are grown in there, and which can be these attenuated viruses, and that can be used as essentially for the antigen or for the vaccine. Now, what is the interesting part, that now the tobacco plants are now modified in the transgenic, where the tobacco plants actually are now expressing influenza vaccine antigen. And you just take these tobacco plants, crush the leaves, and you just isolate these proteins. And these proteins are the recombinant proteins. So we already talked about these vaccines, which are produced in the E. coli or in the yeast cells. But like the hepatitis vaccines are produced, hepatitis A vaccines are produced in the yeast cells or in the E. coli. They are produced in insect cells, like in the baculoviruses, for example. Papilloma vaccine is produced in baculovirus insect cells. But now the recombinant, even the influenza vaccine, is now produced in tobacco plants, so plant origin. And this is the length that where the bioengineering of plants can actually go. So the next part is that we are going to talk about this insecticidal protein, which is a Bt toxin, and the bacteria, which is a Bacillus thuringiensis, that is mildly toxic to the insects. Because the toxin which is present in the bacteria is basically what is called a protoxin, and it is not that toxic to the organism. But what happened that when the sporulation of these Bacillus thuringiensis, is a bacillus is going to produce the spores, endospores, and in the endospores, a lot of these Bt toxin crystals, the procrystals, are crystallized, and this is known as this parasporal crystal. And these are essentially ingested. These can be these could be on the surface of the leaves and all those kind of things. And when the protoxin is ingested, in the alkaline environment of the insect gut, in the arthropod gut, it is proteolyzed by the host enzyme in the form of an active toxin. And the active toxin actually integrates into the plasma membrane, and there are multiple of them, and this basically makes a channel through the plasma membrane of the midgut cells, through which the water ions can leak out, and is resulting in the insect death by the osmotic imbalance and causing cell lysis. So this Bt toxin is produced industrially in large fermenters, so where the Bacillus thuringiensis are produced in these large fermenters, and there is a condition that the sporulation conditions are induced in that condition, where these parasporous crystals are isolated, dried up, and then used in the agricultural as agricultural insecticides. About 75% of the biopesticides are of those, and it is very safe to use because it is not harmful for mammals or animals. There is no side effect of those. So these are usually some of the application. Now, why still one of the problem that can actually happen with the recombinant Bt toxin transgenic plants, because some scientists actually fear that we have not studied the horizontal transfer of the Bt toxin from a transgenic plant to another organism. So the idea is that there are lots of these weeds and undesirable plants. And by chance, if this gene is horizontally transferred from one transgenic plant to another transgenic plant, that can cause problem, basically to eliminate them, and that can cause ecological imbalance. Whereas when we produce the insecticidal toxin, here we are producing the toxin, and that is in the environment. It is naturally degraded in the environment after some time, and it does not accumulate, and it is totally environmentally safe. So this is one of the great application of agricultural biotechnology where we derive a product from a microbe. And this is known as a crytoxin because the reason is like it is derived from a parasporal crystal, and that is the reason that this toxin is known as a crytoxin. So the Bt toxin, there are two types. There is a crytoxin. There is another toxin known as a CYT toxin, which is a cytolytic toxin, which is slightly less active than the crytoxin. These are the two toxins. So crytoxin is nothing but the parasporal, the Bt toxin derived from the parasporal crystals. All right, so after that, we are looking at some microbes are essentially the products, and we are going to take a look at a couple of them. Say, for an example, we know that one of the very important for the leguminous plant is the rhizobia. And these rhizobia basically make these root nodules, which are essential for the nitrogen fixation for the leguminous plant. And this is a direct product where the microbes are essentially the products, and this is also very helpful. So here we are talking about these diatoms, and these are in between about 20 to 100 micron in size. And these are photosynthetic protists, which produce these intricate silica shells, which is known as these frustules. And these are the platform for the biotechnology, specifically for the nanotechnologists to actually have come up with many different types of applications. And due to this controlled pattern and pore size, and these can be genetically also modified as well for some specific characters. So here you are seeing a scanning electron micrograph of some of the diatom, which does have the threefold radial symmetry, but this is like pseudo colored. This is a black and white picture, which is a pseudo color from that. And you can see that there are some very specific geometric pattern and is achieved in this process. So what are the applications of that? So essentially these mineral elements, that is silicon, are incorporated into these biological structures, and these silicon transporters import the element, and the frustule is constructed both through the self-assembly and through the reactions that occur in the deposition vesicle. So very interesting that what happens, that the aluminum, germanium, tin, these could be coordinated with the silicon hydroxide, which is present in the silica, and that can give very unique optical property. And these elements, these structures can be used in solar cells as well. So these are some of the applications. And these diatoms are also used for the drug delivery. So another very interesting part is that sometimes, say for example, at 900 degrees Celsius, these silicons can be actually replaced atom by atom by, say, magnesium. And this is so essentially it gives basically a mold for making a structure, a nanostructure made up of a magnesium instead of the silicon. So silicon is a starting material. From there, it is replaced by the magnesium. Lots of different types of applications. And also used for the drug delivery. They are chemically inert and mechanically very stable. And their large surface area can be modified to conjugate the other molecules. So essentially, some chemical linkers are actually added, and these chemical linkers can be attached with other biomolecules. Say, for an example, one of the classic example for cancer drug delivery, where the vitamin B12 is actually attached by chemical linkers on the surface of these structures, diatoms. And the toxin, which will kill the target cells, are packed inside the diatom, and that can be released in a very slow manner. Now, how this is delivered, that the certain cancer cells actually express high level of vitamin B12 receptor. So when this nanoparticle are injected, they will circulate and ultimately find these vitamin B12 receptors more on the cancer tissues, and these will bind to those receptors, and the drug will be slowly released into the site of the cancer or the tumor. And this is essentially a nanoparticle mediated chemotherapy. And the porous structure will allow the slow release of the drug to the required site. So these are some of the applications in the nanotechnology that holds a lot of promise. The next one is the biosensors. And microbes can sense the change in the environment, and like toxins in water, like a disease process, they can just sense many types of factor molecules, amino acids, a particular chemical. And what we can do is like the linking this microbial detection to environmental changes with an output that humans can actually interpret is used in a biosensor. And in this process, the ribose switches are actually used. So how these ribose switches are used, these ribose switches are essentially, we talked about that, they are transcriptional regulatory elements, and they're found on the 5' end of the RNA transcripts. And this is found on the 5' end of the RNA transcript. And so after this structure, whatever is in the downstream gene, that is not important. But what happened, the RNA folding patterns depends on the binding of an effector molecule. And in one configuration, the ribosome actually, the ribosome binding site is essentially, so essentially it will be an RNA, and this is a transcript, and the ribosome must bind to express the gene. So in one configuration, the ribosome will be able to bind to the, essentially will bind to the ribosome binding site and will express the gene. On the other configuration, it will hide the ribosome binding site in a certain way that the gene expression will not happen. And this property of the ribose switch is essentially exploited in this process. So because the ribose switches can detect amino acid, ions, vitamins, nucleotides, even fluoride can be used in this process. So how this is used, that in a vector, there is a constitutive promoter, and this constitutive promoter, after that there is a fluoride ribose switch, and this is a ribosome binding site, and then it is fused with a green fluorescence protein. So essentially what happens, if the ribose switch is off, then the green fluorescence protein transcription will not happen. If the ribose switch is on, then the green fluorescence protein transcription will happen, and it will produce a color essentially. So in this process, the water sample, so this is the structure. So here water sample are without fluoride, and this is the kind of a structure, the secondary structure of the ribose switch. Now, in this configuration, the ribosome RBS, this ribosome binding site, is hidden, and the GFP is not expressed, and there is no color change in the water. But if the sample is with fluoride, and these water will essentially, what will happen, that when this is essentially detected by the ribose switch, the fluoride ion will bind to a certain point on the ribose switch, on the on a certain sequence, and it will cause a change in the secondary structure conformation, and the ribosome binding site will be now open, and now ribosome can bind, and it can express the GFP, and this can be detected by a green fluorescence. So this is how these biosensors can be developed. So there is a biosensor for heme, and it is developed in animal models. They are not used yet for the human beings, but a very interesting process. So here what we have, that there is a reporter's gene operon is used in this process, and these are LUX CDABE. So this LUX operon actually express the luciferase gene, and the CD are basically the enzymes, and the ABEs are the substrate. So when this is induced, the luciferase actually emit a light. So this is essentially, so one of the example that if you just think about that, where you find this luciferase, where it produces the light, and these are the lighting bugs that you see in the night, and these are that is basically the luciferase gene and the production of light. So now you have right here the LUX luciferase CDAB operon system. And here you have the operator promoter region, and on the operator, our repressor is actually bound. So the LUX CDABE is not expressed. But when a drug like in heme is bound to this repressor, the repressor actually falls off the operator, and now the RNA polymerase is recruited, and LUX CDAB, these gene transcripts are produced, and that will cause a lighting reaction. So the biosensors are made with this construct, and along with that, there is a micro miniaturized electronics is given, so that when in that biosensors, the light is detected, it will emit a signal, and that can be wirelessly detected by a device outside the animal. So the how an in situ testing happened, that you just allow the biosensor, which is a tiny microscopic structure, you allow the animal to ingest, or in future maybe the humans to ingest. If it finds a blood in the intestine, say for an example due to cancer, or say for an example due to ulceration, from that site it's going to emit light, and the miniaturized electronics is going to emit a signal that can be detected outside the animal, and you can have an in situ testing of these. So these are essentially some of the application of that. So these genes are essentially, although the lighting bug actually used that luciferase gene, but this is essentially in many of the bioluminescence in the entire kingdom, which is the eukarya, as well as in the bacteria also do have that. And this gene is essentially isolated from a bioluminescent bacteria, which is a photorhabdous luminescence. And it's a very, very superb transcriptional reporter. And that is essentially that's what it is. So it is the lighting is it's a 490 nanometer is the emission spectra of this. And again, as we said, that the lux A and B gene, the A lux A and lux B, they are produced essentially the luciferase, whereas the CDEs they produce the substrates essentially. And the cells which express these genes will produce the light, which can be used in many types of biosensors. So essentially that's pretty much the chapter, and there are a lot of biotechnology, industrial microbiology is basically trying to answer many different questions, and there is a large field that we did not even talk about, that the whole of the pharmaceutical industry pretty much depend on the industrial microbiology biotechnology. And these scale up of this microbiology starts small, and then it is scaled up to produce many of the products, and they are all of these biological samples. And these are some of the tools so that we find solutions to many of the various problems, as we said, and we can exploit these microbial life by these processes. This chapter by no way is complete. It just tries to give you some of the example and glimpse that where industrial microbiology or applied microbiology is heading to, and some of the examples.